In chapter 4, we're starting to talk about cells and how they build up into each other. So we're going to be talking about tissues. So this is chapter 4, the study of tissues. So one thing to remember with our cells is that cells don't operate independently. So since they need to be around other cells and put together with other cells in order to be able to perform all of their functions, we're now going to put all of those cells together, and cells are going to be put together into our tissues. So this was covered in chapter 1, but when we're talking about tissues, these are going to be groups of cells. And these groups of cells are similar in their structure, and they are going to perform a very specific function. So they're going to be kind of similar, but the goal of them is to all come together to be able to perform a very specific function. So these body cells are all specialized, and as these cells are specialized, they then perform a specific function. And again, the goal is to always be able to maintain our homeostasis. So we're going to have 4 main tissue types. Not 4 main. There are 4 primary tissue types that we're going to talk about today. And then we're going to talk about even more as we get into the different organ systems and organ structures in organism. So the first one is going to be epithelial. Our epithelial tissue is going to cover and maintain our boundaries. Our connective tissue is going to support. Our muscle tissue is going to be responsible for producing movement, and our nervous tissue is going to control pretty much everything. So these are our 4 main types here. So we're going to talk about them a lot, but to give you a little bit of an overview of when you hear the term fixed, section, or stained. When we're looking at these tissues, they are too small to be viewed at the naked eye typically, so you're going to need to use a microscope, a microscopy. This has to be able to be used in order to study the tissue structure, but first, in order to do that, the tissue must be fixed, which essentially means it is going to be preserved. And then after the tissue is preserved, it then has to be sliced. And this is going to be into sections. You can actually see through it. And then it's going to need to be stained. So with this fixed preserved, it actually removes all of the water. Stained is going to enhance the contrast. So that way we can see the different colors and the different structures and all those pieces. So whenever we're looking at something that is a slide or it's a histology, which means the study of tissue, this has all occurred in order to make that happen. Here's just an example of this is what our microscopes in the lab are able to look like. Here's a light microscope. This is going to be an electron microscope, but it gives a little bit more of a 3D. The light just does like the 2D distinguishing, but an electron can actually do the 3D. So, starting with our first type of tissue, epithelial. This chapter is split into 2 sections, so we're going to just focus on epithelial tissue in this first section, and then the second one will cover the other types. So you'll also see epithelia or epithelium. It's just going to be a different ending. These are going to be a sheet of cells, and these sheet of cells are specifically going to cover the body's surface, or they're going to cover a cavity. But there's going to be some sort of covering, essentially. So this epithelium, there's two main forms. You will have a covering or lining epithelium, and because we're only talking about epithelium, I'm going to do a capital E. This is going to be an example of our skin. It's going to be the outer, essentially covering of something. And then you can also have a glandular epithelium, and this is going to be the second part what I'll talk about today. And a glandular epithelium is going to line or create actually the glands of the body. So we're going to talk about those. But overall, our epithelium has some main functions, and these main functions are going to be why the structure and function we're going to talk about as we dive in deeper. are all going to come back to what is our epithelium and why we need it. First one is going to be protection. It is the barrier between two different environments, so it needs to be able to protect. It is also going to be involved with absorption, filtration, secretion, so getting rid of something that is a product, versus excretion, which is getting rid of a waste. And then also sensory reception, so the ability to pick up changes in things and send that signal to the nervous system. So if you think about what we talked about in homeostasis in chapter 1, where we talked about the temperature changing and the sensory receptors in the skin would just pick up that stimulus of the temperature changing. That was found in this epithelial tissue. That was the sensory receptor. Okay, so all epithelial tissue is going to have some general characteristics. So these special characteristics are going to allow the epithelial tissue to have its function that we just talked about. There's going to be 5 main characteristics. First one, polarity. And polarity simply means that all of them will have 2 sides. So you will have, or 2 surfaces. You will have an apical surface, which is going to be the free surface, and then you will have a basal surface, which essentially is going to be the base or the attached surface. So this polarity, all cells are going to have a top and bottom. The apical is the free side, the basal surface is the lower and attached side, and it will attach to a basal lamina. So when we're drawing this, here is my cell. It is going to be, my apical side is the free side. There is nothing that's attached to it. This side right here is going to be attached to something else. And so this will be my basal surface because it's going to be attached. And this right here is actually going to be a membrane, and that's going to be this basal lamina. And this basal lamina right here is going to be the membrane at the bottom, and this membrane is going to be adjacent to the basal surface, and it is a supporting sheet of glycoproteins and cells that underlie, essentially adhere it to the layer below. When this whole thing is put together, when you've got your basal lamina, he is going to be attached to one other sheet of cells underneath it. That's going to be your, let me draw them over here, reticular lamina. When you put these two together, the basal lamina, which is the bottom layer of the cell, and then the reticular lamina, which is whatever it's attached to, this will be the basement membrane. So all epithelial tissues will have a basement membrane, and it's made up of the basal lamina and the reticular lamina. So I got a little ahead of myself because that is actually going to be number 3. So we're going to come to that in a second. So the first one was polarity, but those two actually go together nicely. The second characteristic, well, here you can see there's the apical and the basal. The second characteristic is going to be specialized contacts. And with these specialized contacts, remember these are cells do not live in isolation. They're always going to be attached to each other. They need to work together. So it's going to be how the cells fit together. All of these epithelial cells are going to be fit together super, super, super tightly. So they are going to be connected by tight junctions. So remember what those were from chapter 2. And desmosomes. Sorry, chapter 3. So this is how we're actually going to be connecting our cells, are these two types. So go back and review those if these do not ring a bell. And this essentially is going to keep these cells, essentially to give them like a water resistant or almost waterproof type property. So the third characteristic is going to be supported by connective tissue. So we've actually already covered this. I got ahead of myself. So supported by connective tissue. Since all of these cells are free cells, where there is a free apical surface and a surface that is attached to something else, this thing that attaches it, where it attaches, how it glues together, is going to be through your basement membrane. And so our basement membrane, as a reminder, to go up here, is going to be our basal lamina, which is the layer on the very, very bottom of the cell, on the basal surface, and it will be the reticular lamina that it's connected to, and these two together will make up this basement membrane. But these two types of connective tissues, your reticular and your basement, are going to essentially define the epithelial boundary. So that is super, super, super important. So this will actually separate your epithelium from other tissue. So that is how we know that the epithelia stops and another tissue will actually start. Okay. An example of why we need to know this, so homeostatic imbalance, is if there is cancerous epithelial tissues or epithelial cells, they essentially are going to not be contained by the basement membrane. And so if this is my surface, it's not going to be contained by this and it'll start to bulge out. And that is where you actually will see those tumors formed, or the bumps, or the three-dimensional, especially when it comes to skin cancer. That's where you'll see those, i.e. a mole. That's what they tell you to look for a mole for. Okay, so there's a few more characteristics of epithelial tissue. So the fourth one is going to be that all epithelial tissue is avascular. This avascular will be there is no blood supply to the actual epithelial tissue itself. It is going to get its blood supply by its neighboring tissues. So it still needs the blood supply, but it's going to get it a different way. There's actually no blood vessels innervating this tissue. But it is innervated. So avascular means no blood supply. Innervation means there is going to have a supply of nervous tissue. So you will still have connections to the communication within the body, feeling sensations, hot and cold, pain. So there's no blood, but there is neurons or nervous tissue somehow. And then the fourth characteristic, so that's avascular but innervated. And then the fifth characteristic is they are going to have the ability to regenerate. And this ability to regenerate is really, really important because these are lining the outside surface of something. So they need to be able to reproduce rapidly. As they're lining the outside, think about how much skin cells you lose. You slough off, you go outside, there's weather, there's rain, you take a shower. All of those will essentially cause you to lose this outside layer. So this epithelial tissue needs to be able to reproduce and replicate that rapidly to still maintain that barrier. So they have the ability to regenerate very, very quickly. And here's an example of the cells, since they're exposed to friction, they must be replaced. So that is why we need to be able to regenerate this layer of cells very, very quickly. Okay, general characteristics of epithelium. When we put all of these together, there's going to be two names when we classify tissues, or sorry, to classify epithelial tissue. We're going to do it two ways. The first name is going to be based on the number of cell layers. So the number of cell layers is going to be the first name. So you can either have a simple layer or you can have a stratified. Simple is going to be one layer of cells. Stratified is going to be a lot more. So simple is a single layer, stratified is two or more. So that's the first way. We will classify them as the number of cell layers. The second name we will do it will be the actual shape of the cell itself. So the shape of the cell, there are three options: squamous, cuboidal, and columnar. So you can go with squamous first, And these are going to be flattened scale-like. I like to call them pancakes. Cuboidal is the shape of number 2, and this is going to be a cube. And then the third one is columnar. So these are going to be column or taller than it is wide. So that is how you're going to have these 3 types of shapes. So all epithelial will have these 2 names, and it will be first and second. So as we go, you can kind of see this example. So here is our simple, 1 layer. Here is our stratified, multiple layers of cells. Or you can have your squamous pancake, your cuboidal cube, or your column columnar. So how I like to do this section is you can go through and read the slides, but what I like to do is essentially create a chart for these next little pieces. So what I would do is as you're going, create a chart that looks like this. And it is going to be tissue name. And then it's going to be structure. And then it's going to be function. What does it do? What does it look like? What does it do? And then where do you find it? Location. So as you're going, we're going to talk about structure, function, and location. So this is how I would actually set up your notes, is I would essentially just create a ginormous little chart. So I'm going to replicate that one more time. This is how I recommend you study for this section. It is going to be tissue name, structure, what does it look like, its function, what does it do, and its location. Where is it found? So I'm going to do this as S, F, L. So I would just create a chart after you're done with all this. So first one is going to be simple squamous. This is going to fall under the category of our simple epithelium. So our first one is going to be simple squamous. The structure of simple squamous is going to be thin, 1 layer thick. The function of simple squamous is going to be rapid diffusion. Because they are super, super skinny, they can allow for things to diffuse super, super quickly through. So rapid diffusion, you will find these in your location. You will find these actually in your lungs because you need the rapid diffusion for gas exchange. And then another place you'll find these is going to be your kidneys, where you will need rapid diffusion for blood filtration. So go back, fill in your little chart, give yourself a row, and do the next one. Okay, so here is what simple squamous looks like. Remember, it is flat, 1 layer, and there's a lot of apical surface, so that is about how you get this rapid diffusion into your cell. Okay, the second one is going to be simple cuboidal. Simple cuboidal structure of this guy. He's going to be a single layer of cubes. Ooh, not cubs. Cube-like cells. His function is going to be secretion plus absorption. So we need to be able to move things out and move things in. You will find him in certain ducts in your body, but also in the tubules of your kidneys. So the places that you will need secretion and absorption, your kidney tubules will come up in AMP2. So what does that guy look like? Here is a picture of our simple cuboidal. So here is it's a donut. Here is our apical surface. If you were to stretch this out, they look like this, but it's just in a ring. So that's simple cuboidal. The next one is going to be simple columnar epithelium. So this is going to be a single layer, or one layer because it's simple, of tall, closely packed cells. So these are going to be skyscrapers that are all put together. The function of these guys are going to be also absorption and secretion. So there's a theme with our simple. It likes to be able to move things through the cell. So the places you will find this guy are going to be lining your digestive tract, specifically your stomach and small intestines. You're also going to find these lining your bronchioles or your bronchi in your respiratory tract. It's also going to be lining the female uterine tubules and the glands. So when we're looking at our simple columnar, here's our apical surface, here's our basal surface, and here are our columns all the way down. Okay, so then you actually have another type of cell of tissue that's called pseudostratified columnar. And the pseudostratified columnar epithelium is named because it is fake. It does not actually have stratified layers. So its structure is actually going to be the cells are one layer but they are going to vary in different heights. So they're not all going to be the same height. So if I'm going to have a cell, here's my basement membrane. He's going to be like this, like this, like this. So it's one layer thick, but they're all going to vary in different heights. That gives it this stratified appearance with the nuclei all at different heights. It is also going to be usually ciliated. So ciliated means that it is going to have small little cilia that will come out and essentially help move things across that surface of the cell. So what is it responsible for? It is involved with mucus secretion. It is also going to then be involved with mucus movement. So the best place to kind of see where these are going to be found is going to be the respiratory tract. So this is the structure that is responsible for moving all of the gross crud out of your lungs that essentially you then expectorate to get out of your system. So it is these teeny tiny little cilia that move the mucus along your respiratory tract. Okay, so that's all of our simple simple epithelium. Next, we're going to do our stratified. So I would do the same thing. Give yourself the same chart that we talked about up here. Just now make this one all of my stratified. So you'll see the themes and the structure and the function. So the first type of stratified we're going to talk about is going to be stratified squamous epithelium. So stratified squamous epithelium is going to be built by 2 or more layers of flat cells because these are squamous. Okay, so it's two or more layers of flat cells. I'm going to do a caveat. The free surface, so the top layer, is going to be squamous. But the bottom layers, or the basal layers, the more you get close to the bottom layers, they may be cuboidal or columnar. But it's that top layer, essentially, when you stack everything up, they're going to be flattened cells. The function of these guys are going to be protection. Big, big, big protection mechanism. And the location is going to be any area of wear and tear. So the biggest one is going to be your skin. So this is the type of epithelium that makes up your actual skin, or anywhere that's wear and tear. So yes, you've probably heard of epithelial tissue is the one that makes up your skin, but specifically, it's going to be stratified squamous epithelium that is going to make up the majority of your skin. So here's an example. The top layers up here are your flat squamous, but down here at your basal surface, they may be a different structure. Okay, the next one we're going to have is going to be stratified cuboidal. Stratified cuboidal epithelium. This is going to be usually, only when you find it, it's going to be 2 layers thick, not 2 plus. It'll only usually be 2 layers thick. Its overall function is going to be in glands. So it's going to be involved in secretion of our glands specifically. But the location of this guy is he is very, very rare. So he'll only really be found in these sweat glands and mammary glands, or when it's secreting some sort of product, is stratified cuboidal. It's very, very rare. The next type is going to be stratified columnar. It's on the same side. Stratified columnar epithelium. So the structure of this, it is going to be lots of layers. So it'll be several layers thick because it's stratified. However, in this type, only the apical surface is going to be columnar. So it's going to be similar to the stratified squamous, where the apical surface is going to have that columnar shape. And the function of this is to be able to provide the transition between environments. So it is found when you need to move from one type of cell or one type of tissue to another type of tissue, or in between organ systems. It will be found in transitions between 2 different types of cells. I said areas, but let's just make it official and say epithelial types. So the location of this is everywhere you need to have their transition, but a couple specific ones. In the male reproductive system, in the urinary system, it'll be the male urethra. These can also be found in the pharynx, which is the throat, and then in some of the ducts that we'll have in our system that we'll talk about in a second. Okay, you do have another type of stratified that is called a transitional epithelium. So this falls into the stratified category. And so the structure of this, it is going to be several layers, but it will actually look like stratified squamous. And the basal layers will be cuboidal or columnar. And the top layers will be a little bit more on the squamous side. But this is different than stratified squamous because the goal of this is these are going to be found in areas where you need to have stretch. So that is the function of these guys. So when you've got all of these cells stacked on top of each other, so here's all my cells, they're all stacked on top of each other, they essentially can then stretch out and line one to one. So they have the ability to slide over each other. So the areas that you will need these are going to be actually limited. So not as rare as the other one, but they're not going to be found everywhere. And they will only really be found lining hollow organs that need to stretch. So an example of this is going to be the bladder. Your bladder needs the ability to grow and expand. Urethra and the ureter. All are hollow organs that are going to be lined with this type of cell. Okay, another type of cell we're going to have in our epithelial, those were all of our covering and our lining. These are going to be all of your glandular epithelial cells, or glandular epithelium. So all of those were covering and lining. These are going to be glandular. So what is a gland? Let's define that real quick. A gland is going to be one or more cells that makes and secretes. So it's going to make it and it's going to secrete it outside the system, an aqueous fluid. And specifically, it's going to produce something that we will call from now on a secretion. So like I said, an secretion is different than a excretion. So excretion is the exocytosis of some sort of product that you have created, where excretion, you are getting rid of waste. So secretion is going to be something that has been made. Excretion is something that you're just getting rid of. So when we're talking about glands in the body, we're going to classify these in a couple different ways. 2 different ways specifically. So we will classify our glands based on where they release the product. Essentially, they have produced this secretion, where they release it. There are 2 options for where they can secrete this secretion. They can either secrete it with what is called an endocrine product or an exocrine product. An endocrine product is going to be called internally secreting. Essentially just that means that is a ductless. There is not an opening to outside of the system. If you look underneath your sink and you were to look at the plumbing that is underneath your sink, that plumbing is going to essentially go throughout your house. That is what we're talking about, a duct system. Or plumbing or some sort of, there is no duct leaving once it is secreted out into this system that is going to travel around the body, but there's no essentially duct or door to get out. It is ductless. Or it can secrete, release the secretion that is an exocrine product, and that will be externally secreting. And this externally secreting is going to be outside of the system. So it's going to be somewhere that is secreted outside, away from the system. So this is going to be a duct system. A duct system just means that there's going to be a door. In order to secrete outside, you have to have a door. And then another way that you're going to classify these glands is not where they release the product, but it is going to be the number of cells that are essentially going to form the gland. So when we're talking about these glands, remember we're talking about a bunch of epithelial tissues put together. So you can have a couple different things. Here is going to be an example of, this has a duct. Do you see that? There's a duct, so it is going to be an exocrine. This has no duct, so it is an endocrine. Duct means door. So that's kind of how you can put those 2 categories together. So when we're talking about our endocrine glands, we're going to first talk about those for a quick second. When we're talking about our endocrine glands, these are our internally secreting. The main thing that we're going to talk about is this will secrete hormones. Hormones are going to be the major chemical messengers that will travel through our blood and act on specific organs. So hormones are a big part of how our body maintains its homeostasis, and these are going to be produced by these endocrine glands and then released into the blood supply. This is why they are called ductless, is because there's no good door leaving the blood supply. They are staying inside the system. They do not leave the system. Okay, all of your endocrine glands are going to target a specific organ. So as we go throughout this course, we'll talk about some hormones that will come up a lot. Just keep in mind that a hormone is produced by your body and released into your body, and it's targeting on a specific organ in a specific way. So as these come up, we'll talk about the ones that apply to the systems as we talk about them. So that's an endocrine gland. An exocrine gland. Again, this is going to be our externally secreting. So it is going to secrete the products outside of the system. And with this, when it's outside the system, it could be onto the body's surface, or it could be into a body's cavity. But it's going to release essentially it out of the system. Okay, you have more of these than endocrine. This is going to be the primary one. All of these are going to have ducts. So these are going to have the doors out of the system. So some examples of these guys are going to be sweat glands, oil glands, salivary glands, which are producing saliva, bile glands. These are all examples that are creating some sort of product. Sweat is going to be the secretion, oil is going to be the secretion, saliva is the secretion, or bile is the secretion. So these can be unicellular or multicellular. So when we're talking about unicellular exocrine glands, don't let that term confuse you. It is an exocrine gland, so you know what that means. And unicellular essentially means it's going to be made up of one type of cell. So when we're looking at these, the only important one that you need to know is the mucus cell or goblet cells. And these are going to be in your intestines and your respiratory tracts. So here is a picture of this. It is one cell. It's a unicellular, and it's got a door, so it's an exocrine gland. So it is super easy. It is going to be one cell, and it is kicking things outside of the system. So it just seems crazy when you think of unicellular exocrine gland, but use the terms in the name that you know. The next type is going to be multicellular exocrine glands. So this is also going to be outside of the body. But it's going to have more than one cell because it's multicellular. And this is going to be made up of two parts. There is going to be a duct and something called a secretory unit. The duct is going to be the epithelium, and essentially it's the door, and the secretory unit is going to be however the blood and nervous system is going to be supplied to this system. So this is getting classified in two ways. The first one is going to be the structure of the duct and the secretory unit, and then the second one is going to be the mode of secretion. So for this one, you can get really, really complicated and really dive on deep, but what I want to do is just move on to this image right here, and this is the focus to just kind of understanding what is going on. And then that is, yes, the end of it. So looking at our multicellular exocrine glands, you can classify it two ways. So if you classify it two ways, you can classify it by structure of the duct, which is this way. It can be simple or it can be compound. When it is simple, it is essentially going to be unbranched. So we have two options. Unbranched. It looks like this. See that? There are no branches. Or you can have compound. which is going to be branched. And that means essentially this way, or this way. So here is 1, 2, 3. So that's how you can classify it based on the structure of the duct, or you can classify it based on the structure of the secretory unit. And that can either be tubular or alveolar. So when you have something that is tubular, they're going to form tubules. Tubules essentially are going to be tubes. They're long and skinny. Whereas if you have alveolar, these are essentially going to be bubbles or small hollow cavities is what your book says. But I like to think of them as one is a tube, one is a bubble. So you can have something that is simple alveolar and it is one bubble, or you can have something that is branched alveolar, multiple bubbles. Or you can have a compound. So a compound was essentially, it has the branches, branch, 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 and then bubble, bubble, bubble. Do you see how that is different than branch, 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 tube, tube, tube? There is also something called a tubular alveolar, which means it has both. But you guys just need to know that it exists. So for this, know the pictures, know the structure of the secretory unit and the structure of the ducts. But I'm not going to necessarily give you an image and ask you to show it to me and name it. So just be able to understand the concept between these different types of multicellular exocrine glands. Okay, and then the last part of this section is with these multicellular exocrine glands, so everything that we just talked about before, there is going to be how you actually have the mode of secretion. How do you actually get this product out of the system? So there's 3 different ways. The merocrine type glands, and these are going to make sense. The merocrine glands will essentially get rid of its product because it will it will secrete the product by exocytosis. So as it produces a product, it will just secrete it out of the system. Examples of this are going to be sweat glands, saliva, salivary glands. So as it is secreted, as it is produced, it is secreted. Merocrine. So they are secreted as they are produced. The second type is going to be holocrine glands. And a holocrine gland will essentially accumulate the product. And it will accumulate until it will actually rupture. So if I'm going to have this simple tubular, it is going to fill up with the product until it explodes out, until it ruptures. An example of this is an oil gland. So this is where pimples and whiteheads will come into play. They are essentially a holocrine gland. It's a sweat gland that has gotten plugged up, and it will accumulate the product until it essentially ruptures. And then the third kind is going to be an apocrine gland. And an apocrine gland is going to be similar but a little different. So this is also going to be an accumulation of the product, but it will only have the top layer of skin or the top layer will rupture. Because remember, these are epithelial, so we're talking about the apex layer. This is still a little iffy, but an example of this would be the mammary glands. So the structure of the gland is still intact. It's only essentially released one side of the cell, and that's where you get the mammary gland, apocrine gland concept. So here is kind of an example to show the merocrine glands. It produces its product, and it just releases on out of the system. Whereas in a holocrine gland, it will actually collect it all until it explodes out. So that's the difference, and you just need to know that the apocrine gland is the third time. So that is the first part of chapter 4.